Okay, so let's pick up where we left off. Let's finish chapter 2, shall we? And we'll get hopefully a good start into chapter 3. So we last left off, we're talking about lipids, and we just finished off talking about the nutritional lipids. And now we're talking about phospholipids. So phospholipids are similar in structure to triglycerides because they have the same glycerol backbone. And so in this case, instead of actually having 3 fatty acids on it, it's got what's called a polar or hydrophilic head, a phosphate head that's on top of that third one. So you have 2 fatty acid tails, and you have the polar hydrophilic head on top of it. So these guys are nonpolar, and guys are polar. Now, what this basically does is essentially means that this is a very tense molecule, right? Because the polar world does not like the nonpolar world, right? They hate each other. So here you essentially have a molecule that hates itself to the point where it actually wants to annihilate itself. But it can't do that because it loves itself at the same time. So it's a very disturbed molecule. This tension oftentimes will lead to spontaneous structure. And in this particular case, that spontaneous structure is our lipid bilayer. So this is basically what our lipid bilayer looks like. This is the structure that these phospholipids will take. And the point is basically this area right here, this phospholipid tail, this area right here excludes water. And it gets rid of water. It's like the no water zone. And it points or orients these polar phosphate heads toward the water. So you kind of use your phosphate heads to kind of shield yourself from water, and you kind of create this spontaneous structure, this kind of lipid spontaneous structure. Now, another type of lipid, we're going to talk more about that when we take a look at membrane and the function of membranes. But the other thing we're going to be taking a look at is another type of lipid, steroids. And these are pretty important, especially for human biology, because what typifies a steroid is this 4-fused carbon ring. It's a very classic look of a steroid. And so if you see this 4-fused ring, it doesn't matter whether you know what the steroid is. If you see that 4-fused ring, it's a steroid. And so oftentimes this is a group of functional molecules that have a lot of different functions. The base or the foundation molecule for all of our steroids is cholesterol. And so cholesterol is our precursor that will lead to a lot of our steroids. For instance, we have things like testosterone and estrogen. So both of those are steroid hormones, and they are both basically modified from cholesterol. So cholesterol is an important molecule to have. So even though you have people saying, oh, don't eat any cholesterol, it's like, no, you need cholesterol, because cholesterol is sort of like that foundational molecule that leads to all your other steroids, and they typically tend to be pretty important. Like, for instance, testosterone is our sex hormone, or masculine sex hormone. And testosterone is potent. Most steroids are. They're very potent. And so testosterone is essentially what masculinizes body tissue. Like, for instance, when we're all kids, before puberty, before our steroid hormones kick in, a kid's body is kind of the same, right, between boys and girls, and for the most part. And then what happens is puberty hits, testosterone kicks in, and that masculinizes the male body into typically the adult male, which is larger muscle mass, hair, those sorts of things. And estrogen will feminize the body. So that'll basically create all the secondary female sex characters, breast development, the shape, right? The shape of female muscles is very different from male muscles, right? And so we kind of see that in terms of just kind of the shape that the body takes on. And so the female body tends to be much more kind of aquiline in nature, whereas the male body is a bit more kind of bulky and clunky, if you will, in a lot of ways, right? And so that's estrogen doing that. And so they're very potent hormones in the human body, and so they're necessary. So that's the reason why you don't want to just have a diet with no cholesterol in it. It's also important as a component of your membranes. So your cell membranes need cholesterol in them in order to stabilize those lipid bilayers. So you do need cholesterol. You just don't need a ton of it, right? And so it's kind of what it looks like. So here's your cholesterol molecule, and you'll see you basically have this 4-fused ring. And so any molecule that has this 4-fused ring is a steroid. And this is also important, like especially if you get like a prescription and you ask the doctor, it's like, oh, is this a steroid? Don't ask the doctor; they don't know half the time. Right. So just ask them. When you go to the pharmacy, just ask them for that little pamphlet that they give you of information for the medication. They have to give you that, right? Because that has all the side effects and all that sort of stuff. But it also has the chemical structure of the medication in there as well. If you take a look at the chemical structure and you see this 4-fused ring, it doesn't matter what the doctor says. It's a steroid. Okay. Now, from cholesterol, you can modify this into testosterone and estrogen with just a few modifications. For instance, this large blue branch right here, you can basically take off and swap it out for an OH group. And instead of this little OH piece over here, you can basically put a carbon double bond. And that's pretty much it. And with, of course, a shifting of this bond over to here, that's pretty much it. So those are the only modifications you make when you go from cholesterol to testosterone. So very, very small, very slight modifications. It's even more amazing when you compare testosterone to estrogen, right? Because as estrogen, you still have this OH group. That's also the same with testosterone. You kind of miss this methyl group there, and you keep your hydroxyl group, and you basically have alternating double bonds on this end piece. And that's about it. Those are the only differences separating testosterone from estrogen. So it looks like some pretty minor changes to the molecule leads to very titanic differences in function. And that's kind of typical of steroids. So that's the end of your lipid section. So now we're going to be digging into the protein sections, hopefully coming to kind of our little bit of a home stretch a little bit. And so the first thing we're going to be taking a look at, because proteins, we've already covered carbohydrates. We haven't covered nucleic acids yet. But the next one now is proteins. Proteins basically do a lot of different things. Pretty much if there's a biological function in your body, it's a protein that's doing it. And so they have lots of functions. They'll do like support, for instance. That's a big one. So for instance, keratin, which forms hair and nails, that typically will support protect your nail bed. It's kind of for an animal, for a mammal, it's claws essentially, or fur. Collagen, which is also an important protein. That's a supportive protein. That's basically the main protein in ligaments, and also in the underlining of your skin itself. So the dermis, the actual leather portion of your skin, is actually mostly collagen. They actually are catalytic in nature, which basically means they speed up chemical reactions. So a lot of those chemical reactions in your body are being driven by enzymes. Those are proteins. They also will be involved in transport, right? So basically this is regulating how molecules basically enter and leave the cell. So who gets to come in, who gets to go out? Those are transport issues. And so those transport molecules oftentimes are in the membranes, but they can also be free floating in the blood. So you have transport proteins also in the blood that are helping you to transport oftentimes lipids. So there's a lot of different functions that the protein does. And so that's kind of our short list, but we continue the list over here because you also have defense, right? For instance, antibodies typically are proteins. Those are floating around in your blood, and that's essentially associated with your ability to go after antigens, which is any kind of a molecule that creates an immune response, like a bacteria, for instance. So these will go after bad guys. Hormones. You got lots of hormones in your body, not just the steroid hormones like testosterone and estrogen, but you also have a lot of protein hormones like insulin, things like that. These are basically chemical messengers that will help to coordinate the activities of your cells and your tissues across your body into a coordinated global multi-system response toward a particular situation that's happening in your body. Also motion and movement. For instance, muscle contraction. Pretty much your muscles are just about almost all protein. So there's a lot of protein involved in muscle contraction, and also how different sorts of things move within the cell itself. So how do you build proteins? Basically, you have the same kind of situation where you have the fundamental, those are just motion censored, if you're wondering why they're going on and off. So basically, they're built by the same process, right? So you have monomers, or the individual subunits called amino acids. And so an amino acid is very basic, has a couple of basic pieces. First of all, it's got the amino group. So this is your amino piece. And then in the middle, it's got what's called an R group carbon or a alpha carbon. So it's kind of a hydrogen attached to one side, and it's kind of an R group, which you have 20 different versions of that R group to choose from. And then you're going to have your carboxylic acid. So this is going to be carbon that's double bound to an oxygen, single bound to an OH group. So this is your acid group. So amino acid. So you have these 3 functional groups. All amino acids have that same structure, that same backbone. What differs between them is their R group. So they have the same basic adapter. They just differ in terms of the R group that they have. And so here's kind of like your amino acid. Here's your amino group, your acid group, and in the middle, you've got your R group carbon. Now, there's different types of amino acids. We're not going to go through all of them, but this is an example of some of them. For instance, some of the amino acids are nonpolar, like valine. And because of that, they'll have a particular personality, a nonpolar personality. Some of them are ionized, that is to say that they're charged and polar. For instance, glutamic acid has that negative charge sitting there, and that gives it a particular type of personality. Some of them are negative charge, or some of them are positively charged. So this is an example of a charged polar amino acid that has a positive charge on it. Tryptophan is another type of a nonpolar, but in this case, it has a bunch of rings in it. And then aspartic acid is another type of an acid, so that's a negative charge. You have something like cysteine. So you have basically polar and nonpolar, charged and uncharged, ring or no ring. Those are the main structural motifs that you have. And then you have cysteine, which is kind of a bit of an outlier because that's got this sulfhydryl group or the sulfur group on it. And those are usually very specifically doing something very specific. So they allow you to cross-link proteins as they're folding up, and that basically stabilizes the protein structure. So when you're putting these two guys together, you create a bond, a covalent bond, called a peptide bond. And so this is the bond between the two amino acids. As you build more and more of them, then you'll create a polypeptide, which is a long chain of amino acids. So it's kind of what it looks like. So, oops, there you go. So here's one amino acid and the second amino acid. So they have different R groups. So this is R group number, I'm going to say R group number 1, R group number 2. Remember, whenever you do a dehydration synthesis, you have to have the components of water, right? And so the OH of the acid group on one amino acid is the OH donor for that water. And then the hydrogen on the amino group of the other is the donor for the rest of the hydrogen. So what's going to happen is your enzyme is going to cut this bond here, and it's going to put these two together to form water. So you're going to exit as water. And then now that you've done that, you have to join these two monomers together. So it takes the acid carbon of one amino acid and connects it to the amino nitrogen of the other. And that becomes the peptide bond. So when you form the dipeptide, this one right here, which is connecting the acid carbon of the acid to the amino nitrogen, is the peptide bond. So now what you have is a dipeptide where you have 1 and 2 R groups on it. Okay, now all you do is just keep on adding that in your dehydration synthesis. So you're basically just taking this one and then adding the next amino acid to that. So the forward reaction is our dehydration synthesis. If you want to pull these proteins apart, then you just do the reverse reaction, hydrolysis. In this case, basically, you just cut the peptide bond into 2 separate amino acids, and then you can use that for whatever you want to use it for. So proteins are important because for them, when they're doing their job, 3-dimensionality equals function, right? So for them, 3-dimensional shape equals function. And so it's very important that they maintain their 3-dimensional shape. If they don't, then they're not going to be functional. It's just that simple, okay? So one of the ways that you can destroy that shape is through a process called denaturation. Now, denaturation itself is essentially the unraveling of a protein to a shapeless, non-functional form. So you can do this in one of 2 ways. First of all, you can use extreme heat. Or you can use pH, right? So with extreme heat, the example here that I like to use is like when you cook an egg, right? So when you crack an egg into a frying pan, or actually just when you crack an egg open, what color is the egg white? raw? Not a trick question. It's clear, right? So that is the protein, mostly that's in egg white is a protein called albumin. And it's ubiquitous in animals. So we have a lot of albumin in our own blood plasma, and it's in amniotic fluid. So it's pretty much everywhere. Then when you basically put that egg into a frying pan, what does that egg white look like? It's white, right? It's opaque and white. You can't see through it anymore. What's happened there is you've used the extreme heat of the frying pan to basically denature the protein. So the proteins, which are three-dimensionally folded up into this three-dimensional shape, basically just the heat just kind of melts it, so to speak. It unravels, and then all those proteins get entangled with each other like a really bad set of Christmas lights, right? And then they're irretrievable at that point. So that's what creates that sort of opaque gamish of protein. So that's just basically a big wadded tangle of proteins. That's what the egg white is. Okay. And so that's denaturation. You can do the same thing with pH. For instance, you can basically make cheese with lemon juice. Have you ever made cheese before? It's a very simple process. Usually you make cheese. The first thing you have to do is get very fat and protein rich milk, like whole milk, for instance, or raw milk is the best, right? But the idea is you usually use an enzyme called renin to denature all those proteins that are in the milk, and you kind of start to create curds and whey, right? Which is basically a fancy way of saying what? So you guys have all eaten curds and whey, probably. So what do you think curds and whey is? The curds are the solid pieces that clump together. The whey is the liquid. Maybe you like a good bowl of curds and whey, right? The nursery rhyme, little Miss Muffet sat on her tuffet eating her curds and whey. You know what she's eating? Cottage cheese. So the chunky parts of the cottage cheese are the curds, and then the liquidy part is the whey. That's the beginning of cheese making, essentially. In this case, you have to denature those proteins in order to get those curds to clump up and to coagulate. Now, a lot of people don't like the renin use because it actually comes from intestines. It's an intestinal sheep gut enzyme, and so they'll make vegan cheese, although it's still a milk source. But anyway, but they'll use lemon juice to make it instead. And so in the acid environment of lemon juice, it has the same effect. It'll basically cause all those proteins to unravel. They'll start to cross-link and get tangled up. They'll start to trap all the milk fats along with it, and you kind of start to coagulate into these curds, and you have the liquidy part. Now, in order to make cheese, basically what you do is you take cheesecloth, you sort of capture all the curds. You actually do use cheesecloth for making cheese. So it has the thing. It's not just called that. Right. And then you put it on a cheese press and you basically squeeze out as much of the water in the liquid as you want, depending how dry you want your cheese. And then you wrap it up, give it a brine wash or a bacteria wash, depending on how you want to age it. And then you can just set it in the dark and just let it do its magic. In a few months, you got yourself some cheese. The longer you let it sit, the longer it ages, the sharper the cheese gets. So those mild cheeses are cheeses that haven't aged for very long. Those really sharp cheeses, those are cheeses that have aged for a long time. Okay, so they get sharper as it ages more. But that's an example of cheese making, right? With basically denaturation. You're denaturing everything, and it's just kind of falling apart. You've also seen this effect. Some of you perhaps have, when either you or somebody you love, like a kid usually, happens to make the mistake of drinking milk when they've got the flu, which I still have PTSD from, because I have a hard time. I love cottage cheese. I have a hard time eating it because I still have flashbacks. Because there was one flu season where all three kids got the flu, and that was the season when we went from carpet to all wood in the house, because at some point there's only so much Stanley steamer can do. And so yeah, we were just like, no, not again, no more. We lost chairs, we lost couches. It was, the kids just can't, they don't make it to the bathroom, right? They just, like, if you're there and you're holding them, it's just like all over you. And this is kind of what happened. I made the mistake because my daughter was getting over her cycle and she was getting hungry. So that was a good sign. So I'm like, she wanted some milk. So I'm like, okay, I think you're over it. Of course, what happens? Worst decision ever. She drinks the milk. And then here it all comes, right? Like, here it all comes. Why? Because when kids don't feel well, who do they go to? They go to mom and dad, right? And then, of course, you're taking care of them, you're holding them, and then here it all comes. And it's just like, that was my wife. She was the one who got to wear it all. I was the one, while she was getting them in bathtub, I was the one who was lucky enough to be able to stay behind and clean it all up. And there's nothing worse in life than trying desperately to get half digested milk that has come out of your stomach, which has a pH of 2, same as lemon juice, by the way. Right? And when that comes up, you've got all these little curd chunks that come out because of the denaturation. And imagine now it's all over you. The kid is in the bathtub because, I mean, just getting hosed down because there's no other way to do this. And you're basically trying to take your shirt off while at the same time trying not to get everything that is on you basically smeared all over your face and up into your mouth and into your nose while it's coming off. We haven't even gotten to the smell yet. So I feel like I have a pretty strong stomach, but there's a couple of things that will push me to the edge, and that's one of them. Half digested vomit milk is one of them that gets me right on the edge. So you can see how that this is still a lot of PTSD because I can still remember that very clearly. And to this day, I have issues with it. But that's the same thing, right? Your stomach is acidic. And so it's denaturing the proteins. Right? That's what its job is to do. Right. So now let's take a look at what they're supposed to do when they're not being denatured. So protein has to adopt its three-dimensional shape, right? So it goes through levels. First of all is what's called primary structure. The primary structure of a protein is essentially the linear chain of amino acids as they're being put together. Then what happens is they start to fold into what's called secondary structure. And basically, this is what's called localized folding. This is essentially the backbone of the polypeptide seeing itself and folding on itself. It's just a backbone thing, right? And so it forms two different major types of structures. It's called alpha helix, which is kind of like a little corkscrew shape, or it forms beta pleated sheet. This is the other one, which basically is this kind of like accordion-like sheet that's held together with hydrogen bonds. Okay, so those hydrogen bonds, remember we said are attractions between the positive hydrogens and the negative oxygens of molecules. So this is kind of what it looks like. So here's your primary structure. So you can see just a linear chain of amino acids, and this is your secondary structure where you form your alpha helix. You can see if you kind of blow this up a little bit, you can see how you have this hydrogen bond that's holding this corkscrew, this alpha helix together. So you have the slightly positive hydrogens, basically giving yourself an electrostatic attraction to that slightly negative oxygen. Same thing with the beta pleated sheet. You can see your slightly negative oxygen being attracted to that slightly positive hydrogen. So it's forming a hydrogen bond. So both these are hydrogen bonds holding these two together. The next level is what's called tertiary. So in tertiary, basically, this is essentially where the R groups interact with each other. Right, so remember we just said that the R groups can be polar, non-polar, they can be charged or non-charged, they can be ring structures or non-ring structures. And so depending on what they are, they interact with each other. For instance, non-polars hate polars. So these will tend to run away from each other. And as they run away from each other, because they're tied to the backbone, they bend the protein. So for instance, if I've got a polar amino acid here and a non-polar one here, what's going to happen is they're going to run away. So they're going to try to get away from each other. But as they do that, they're going to bend the backbone and they're going to bend it so that they get as far away from each other as possible. So that's what's happening in tertiary. And so generally speaking, we have all different types of bonds. So we have ionic, covalent, and hydrogen bonds, all kind of holding this tertiary structure together into what basically looks like a big globular protein that, if you take a look at it, is made up of secondary structure. So there's sheets and helix in there, loops and things like that. Then the final level of structure that we're going to see is quaternary structure. This is basically where you have a combination of subunits that are each basically folded up into its tertiary structure. So essentially, quaternary structure is essentially for proteins that function as a team. So a good example is hemoglobin. That's always the one I use, because hemoglobin has what's called is a tetramer, because it has 4 different subunits. Each subunit itself is made up of a tertiarily folded subunit, right? So each subunit is folded up on its own. So it has 2 alpha subunits, 2 beta subunits. And so each one of these is in tertiary structure, but they're not functional by themselves. They only function when they're together as a team. That's quaternary structure, right? So this is typical of complexes. This is what it looks like. So here's your tertiary structure. So you can see how it's kind of a wadded up mess. And you got a lot of different things going on here. So for instance, you've got some loops. That's kind of looks like it's in just linear. You got some things right here, right? These are sheets. You see a little helix right there. See another little helix right there. There's a nice one there. And so you kind of see this combination of helix and sheets and loops and things like that in tertiary structure. Then in quaternary structure, you can see that you basically are a member of a team, and you're only functional as a team. So here you can see you have 4 separate subunits that act together as a team. That's your functional enzyme. So that's quaternary structure. Okay. So last of the major groups, the nucleic acids. Let's take a look at these guys. So nucleic acids, these guys are going to be the informational molecules. So they're polymers of the subunit of nucleotides. The nucleotides have 3 main pieces, very similar to kind of like the way amino acids had 3 main pieces. Only in this case, your main pieces are you have a phosphate group on your nucleotide. You then have a 5-carbon sugar of 2 potentially different types. And the last thing you have is a nitrogenous base. Okay. Now, generally speaking, the main task of nucleic acids is for information storage. And so the information that they have essentially is what you need to basically tell everything else what to do. So all the instructions necessary for all these proteins to be made, for all these enzymes to be made, for all these transport molecules to be made, all that is encoded in your informational molecules in your nucleic acids. So they basically are the sort of the basic blueprint, if you will, right? So we oftentimes hear of DNA, we talk about DNA as the blueprint of life, right? But oftentimes, how many of you guys have heard that one before, right? DNA is the blueprint. The problem is usually when people say that one, they don't elaborate on what that actually is. They assume that's like, oh yeah, you know what a blueprint is. I don't know, I'm not an architect, and honestly, we don't do blueprints the way we used to. And most of the time, most students have no clue, other than just superficial, what a blueprint actually is. They just know it's a set of plans, right? But a blueprint, if you've ever taken a drafting class, and I took one actually in high school, I was terrible at it, loved it, but I was terrible at it because my penmanship, right? You have to be very, very neat penmanship to do drafting. And even on my best day, I was a woeful embarrassment to the establishment. But the important thing is, like, whenever you have any kind of a structure, there's a design or a plan for it, a blueprint for it. And the thing is, the blueprint isn't just a list of how-tos. It's not like an instruction sheet for some sort of a toy or a bike or something that you just got from Amazon. A blueprint is different, right? A blueprint basically has every scrap of information that's necessary in order to build that thing, right? So any little question you have about building a structure is in the blueprint. It's all got to be there, right? There can't be any questions whatsoever because it's all got to be in that blueprint. So it's an exhaustive source of everything you could possibly conceive of that would be a thing, right? So when an architect is drawing out a blueprint, they have to think about all the potential things that the workers could ask. It's like, okay, so you want me to pour the footing here, but how deep and how wide and out of what material, right? And so how far away from the third footing, right? So all of these details, all these questions that you could potentially have to be answered in the blueprint. It has to be an exhaustive source of information. And that's basically what these nucleic acids are. They're an exhaustive source of information to essentially build you. So here's your nucleotide. So here you can see your phosphate group here on the 5' side. Then you can see your sugar piece and your nitrogenous base. Now, one of the important things that we see here that I want to just sort of decode for you is that in organic chemistry, this is an organic chemistry thing. What they do when they're looking at complex molecules, in order to differentiate which carbon they're talking about and referring to, they number the carbons. And that's what these numbers are. So for instance, this is the 1' carbon, the 2' carbon, the 3' carbon, the 4' carbon, the 5' carbon. And so that just basically allows us to be able to differentiate which carbon we're talking about. So when I talk about a 5' phosphate, for instance, this guy over here, then what we're talking about is the phosphate end that's attached to the 5' carbon. Okay. When I'm talking about a 3' OH, I'm talking about an OH that's attached to the 3' carbon. So these numbers basically allow us to be able to sort of navigate through the molecule and be able to sort of understand where we are, sort of map-wise throughout the molecule itself. Okay, so there's two main types of nucleic acids that we see. The first one is DNA, deoxyribonucleic acid. This is your ultimate source, right? This is your ultimate source of information. And so it'll have everything it needs, everything you need in order to make you from scratch, from zero. It is also important because this is where the information comes to build your proteins, right? So for instance, remember when we looked at the primary structure of proteins, we had an order of amino acids. Every time we select an amino acid to put in next, we have 1 in 20 decisions to make, right? So how do we know, for instance, that the sequence of this protein is alanine, phenylalanine, lysine, right? How do we know it's not some other sequence? Well, what tells us which amino acids to pick? It's the DNA. It's all encoded in the DNA. The DNA tells us exactly which amino acids to pick. Okay, and that's in the sequence of the DNA. Now, there's another type of nucleic acid, that's ribonucleic acid, RNA. And so this has several different functions, but one of them basically it serves as a copy of DNA. So this is kind of like a local copy of the master. Think about it. If you're a worker at the site and your job is to pour the footings, you need information, don't you? You need to know where to dig the footings. You need to know where to pour. You need to know what types of things to put in there, right? You need to know where to locate them. You need to know like how to level them. Are you taking a look at a level? Or are you looking at a footing that's kind of like got multiple levels where you're going to have like a different type of kind of a building on that with multiple like different levels or different main levels on it, as like if you're dealing with a hill or something like that, right? So what you need is instructions to help you pour your footings. You don't care about like all of the touches. You don't care about how you're going to build the rooms or all that sort of stuff. That's something that's for somebody else. That's for the carpenters to worry about, right? Your job is just the cement. Yours is the footings. That's what you don't need everything in the master list. You just need your job. So what you typically oftentimes tend to do is at the works, if you've ever been in masonry, it's a pretty messy place, right? So you got mud and cement and all sorts of stuff all over the place. You don't take your master instructions to the work site, do you? Because if you pour a bunch of cement all over them, guess what? You're screwed. That was your master copy. Guess what? It encased the cement now. Good luck, right? So no, what you want to do is you want your information, but you don't want to sacrifice your master. So what you do, you make a local copy. You make a copy that's just focused on your job, pouring the footings, and that's what you have at your work site. So you have a copy of the master blueprint that you can rub your donut on, spill your coffee all over because it doesn't matter. It's just a local copy. We can run another copy off for you, right? And so that's what you have at your site. It's just focused on what you need to do the protein that you need to make at the time that you need to make it. But your master stays nice and protected in the architect's office. Okay. So, for instance, RNA can basically be the code that will allow you to be able to bring that information for amino acid selection to the site of productivity. It can actually act as coenzymes. There are catalytic RNAs that we've learned about and discovered that can kind of help to regulate different types of chemical reactions. A non-informational type of nucleic acid, which is an important one, is ATP, adenosine triphosphate. This is basically our energetic currency. So this is like our rechargeable battery. Whenever we want to do something in the cell, like we want to do some sort of a task in the cell, it requires energy to do it. Where do we get our energy from? ATP. It's just like whenever you want to do something on your phone, you want to work on an app, what do you need in order to get that to work? You need some bars on your battery, don't you? Right? So if you don't have a full battery, then you're not going to be able to utilize that app. And that's how ATP is, right? It's also rechargeable, just like your cell phone battery. So whenever our battery runs low or we use the energy in our battery, what do? We plug it in and we recharge it. And then we go use it all up, and then we plug it in and we recharge it, right? So that's kind of a constant cycle. ATP is essentially your rechargeable cell phone battery for the body. Whatever things that you need energy for, you just get an ATP molecule, and that'll give you some energy, and you go have to recharge that in another process. That's cell respiration. That's metabolism. So here's kind of a construction, a kind of a side-by-side comparison between the two major types, DNA and RNA. So for instance, the sugar that they use is different. In DNA, they use what's called deoxyribose. And at that 2' carbon, what it has is an OH group attached to it. Excuse me, not an OH, it has, erase that one, I'm backwards, getting ahead of myself. It has an H attached to it, so it's deoxy. And in ribose, at that 2' carbon, it has an OH, so it's oxy, right? So that's basically the difference between those two ribose carbons. Bases in DNA are adenine, these are the nitrogenous bases. So there's 4 different bases that we have: adenine, guanine, thymine, and cytosine. So those are basically our A, G, T, and C. In RNA, we have similar. We have A, G. Instead of T, we have U, and then C. So we swap out T for U in RNA. DNA is double-stranded with 2 base pairs. So 2 bases kind of hydrogen bond to each other to form your double strand, whereas RNA is single-stranded. So it's a little bit different. Generally speaking, DNA is a helix, RNA is not. It's straight, it's linear in nature. So those are some of the major differences between some of those. For instance, when you take a look at the structure, DNA will form this kind of alternating phosphate sugar backbone. We'll take a look at that here in just a second. And then from the sides of the backbone, you'll see these bases projecting. And so you have to identify those and sort of pair with those. We'll take a look at how that looks here. In the double-stranded DNA situation, we essentially have a double helix that's twisted, and it's held together with hydrogen bonds through what's called complementary base pairing. So for instance, every adenine forms 2 hydrogen bonds with every thymine, and every cytosine forms 3 hydrogen bonds with every guanine. Okay, so those are the pairings. So when you make your DNA molecule, it's kind of what it looks like. So you can see here, as you sort of put this together, you can see your phosphate, sugar, phosphate, sugar, phosphate, sugar backbone. So this is your backbone. And then to each of the sugar sides, you see these little protrusions of these nitrogenous bases basically sticking out. Now that's basically just one side, right, of the DNA. So you'll see your backbone with these bases. So let's say we have A, C, G, T, A. Now what you do is, in order to make the other strand, you have to pair them together. For instance, your adenines and your thymines will basically form 2 hydrogen bonds to pair each other together. What that means is, on the other strand, the adenine will form two hydrogen bonds to pair with its partner, which is thymine, and this is attached to its backbone. Cytosine will form three hydrogen bonds with its partner, which is cytosine and guanine, right? So that's basically a base pair. So wherever I see a cytosine, I'm going to be forming three hydrogen bonds with the guanine. So cytosine guanine forms three. Guanine will basically form three with the cytosine. The thymine will form two with its adenine. The adenine will form two with the thymine. It's like a ladder, where the rungs of the ladder are sawed in half, but they're held together with Velcro, right? So imagine that, a ladder, a wooden ladder that's sawed in half down the middle of the rungs. And instead you've got Velcro on the ends that holds the ladder together. That's like the hydrogen bonds holding the ladder together, okay? In this case, it's not a wooden ladder. It's like a rope ladder, and it's a twisted rope ladder. So now all we have to do, we have these two pieces together, zippered together with hydrogen bonds. All we have to do is give it a bit of a twist. And when we do that, we create our DNA molecule or double helix. So it's like a twisted rope ladder. And that's a DNA molecule. So you can see your double helix here with your AT base pair, your CG base pair, and so forth. Now ATP is your energy currency. So it's really important. It tends to cycle back and forth, right? So basically you start off with what's called, so here's your ATP. So here's your, what's called your adenosine. So it's what's called a nucleoside. So this is an adenine there that's attached to your sugar. And then to that is attached not one, but three different phosphates. So it's a triphosphate, right? And so basically you have an adenosine that's attached to three phosphates. So that's your adenosine triphosphate. And then what happens is it'll hydrolyze, notice, with water. You have to add some water because it's a hydrolysis reaction. What'll happen is you'll pop off this terminal phosphate, that's this guy right here, and along with that, you're going to get a big burst of energy. And you can use that energy for biological work. What's left behind is adenosine diphosphate. And then so during metabolism, what you do is you have to re-energize this, basically recharge it. And so during cell respiration, you basically go through a dehydration where you basically put a phosphate back onto that ATP and you regenerate ATP, okay? So you have the cycle. You take ATP, then you hydrolyze it, you lose that phosphate, and then you get a burst of energy from that. And then during the recharging period, you take an ADP, which is the leftovers, and you put a phosphate on that to make ATP. And you basically just keep cycling back and forth and back and forth. Okay. So there's a lot of energy in these phosphates, and that's where the energy is coming from to basically fuel the activities of your cell. Okay. So it's a really, really important molecule. So that's the end of 2, finally. But I didn't want to rush that one because there's a lot of important stuff happening in chapter 2 that kind of forms our foundation for some of the things that we need to have in place before we start moving into some of the human applications. So let's take a look now at chapter 3. So we're going to take a look at cell function. And so the first place we're going to start is the cell theory. So the cell theory basically is defined as the basic unit of life, which basically means that this is the smallest we can get and still answer all the big biological questions of life. So that's number 1, right? So it's a basic unit of life. All living things, we noticed also from all of our observations through hundreds of years of observations or microscopy looking at living things, is that pretty much things that we are pretty comfortable with being alive which it was a process of how to define living and non-living. Those things that we are pretty comfortable with being alive, all of them, without question, those are all made up of cells. Okay, so everything that's alive is made up of cells. And the third one was basically answering the question like, okay, so that's fine, I'm good with that. But now, where do cells come from, right? So cells basically come from pre-existing cells, which kind of makes a lot of sense, right? Because think about it, where do puppies come from? Yeah, dogs, right? Where do kittens come from? Yeah, where do babies come from? People, right? So it makes a lot of sense. Cells come from other cells. So there's a sense of lineage and heritage associated with it. So one cell kind of gives rise to other cells. And that's basically the cell theory. And so this is kind of what it looks like. This is an example of some cells. Like, for instance, you can see red blood cells here. These are electron micrographs in a blood vessel. It's kind of cool. This is an osteocyte, so that's a bone cell. And these are nerve cells. So you can actually see that's an actual real image of a real nerve cell. So these are some pretty cool images, some cool electron micrographs. So let's take a look at the function of the cell in terms of what it has to do, right? Because if this is the smallest that we can get and still answer all those big biological questions of life, then it's got a lot of work that it's going to do, right? You have to have a lot of productivity. So the question is, when you're trying to be productive, one of the things you always learn, especially when you look at like from a business angle, right? So you always want to maximize your productivity while minimizing your costs, right? It's kind of a cost benefit analysis. Anybody ever go through one of those? You try to get maximum benefit for minimum cost. In this case, maximum benefit for a cell is given to us by surface area. So surface area basically is our working surface. So the more we have, the more productive we are, right? So the increase in working surface means we have more productivity, we can get more accomplished. However, that comes along with volume. Volume represents the cost. Okay, so that's just how big are you? That's how much you're going to have to invest. Now, generally speaking, when you take a look at this, if you want to increase your working surface, your surface area, it makes a lot of sense sometimes to say like, well, okay, I'm just going to make a bigger cell, right? That'll increase my surface area, and it will. But remember, it's not just about increasing your surface area. It's about what kind of deal you're getting for that, right? So is it justify the cost? Yeah, sure, you're getting more working surface. But are you paying a little more than you should for that in terms of cost? And so your cell is always trying to balance this, just trying to get a larger amount of surface area relative to volume. It wants maximum benefit for zero cost. That's really what it wants, right? It wants to go to a yard sale and it wants to get something brand new for nothing, right? You go to these yard sales and it's like, I'm going to get rid of it. Just take it. Like, okay, I'll take it, right? It's the way I shop for my bikes, right? So I never buy my bikes new because there's just too many people getting rid of their bikes, and I'm not picky. So I don't need like, the upper level perfect models and, the newest technology. I'm good with the 78 Oster Dynamo. That's what I'm riding today. That I get for $20 at a yard sale, right? Which is like really shape. And that's just like, I'm kind of shaking my head. I'm like, okay, if you part with that for $20, I'm more than happy to take it off your hands for $20, right? Or my giant mountain bike that I ride on Fridays that I got for like $40 at a yard sale, which is something more on $400 or $500 range. So that's the way I shop. I'm looking for maximum benefit for minimum cost. This is the reason why I don't go to bike shops, because you're going to be parting with tons of money, but you're not going to be getting that much benefit. So you're not getting a good deal when you're buying that same giant for thousands of dollars because it's new. When bikes tend not to age very quickly, especially if you take care of them, right? So they can be around for decades, like my 78 Oster Dynamo, which I love. Anyway, right? So the idea is, how much benefit can you get for your volume? So if you increase your surface area, you can do more stuff, right? For instance, you can get more nutrients in the cell, get more waste out of the cell, things like that. But ultimately you don't want to get big to do this. Here's a comparison that we have. For instance, this is just a generic, just to show you the relationship between surface area and volume. So there's basically, we're gonna take a look at a cube that's 4 centimeters by 4 centimeters by 4 centimeters. So when you take a look at this cube, the surface here is the working surface area, right? And so when we calculate the surface area, what we get is 96 centimeters squared worth of surface area. Not bad, right? Its volume is 64 cubic centimeters, okay? And so the ratio to that is 1.5 to 1. Now let's compare this by being smart about it. So rather than just getting big, let's actually do some manipulations to increase surface area without substantially increasing our cost or volume. So what we're going to do now is we're going to basically cut these squares in half. So we're going to cut this basically in half like this. So you're going to create these like little squares. So each of these now are surface area. And also in here and in these little nooks and crannies, you can't see them, they're kind of scooched up next to each other. But now you've got all these different surface areas in there. And so what you've done is you've increased your surface area to 192 cubic centimeters. Now, if you cut these guys in half, then you get even more little cubes in there. And so all these little surface areas add up are 384 cubic centimeters. But notice what happens to your volume. Your volume pretty much stays the same. You didn't get bigger, you just sort of subdivided yourself up, exposing new surfaces that weren't exposed before, allowing you to now utilize a new set of surfaces to do work on. Okay. So the ratio for the 8 2-centimeter cubes is 3 to 1, doubled your productivity, right? From 1.5 to 3, but you maintained your cost. That's pretty smart economics. If you can double your productivity and maintain your cost. And then when you divide it up again, you doubled it yet again, but you maintained your cost. So you're basically increasing your surface area while minimizing your volume or your costs. Okay, that's exactly what you want. You're getting maximum benefit from minimum cost. Now, the reason why we mentioned that is because cells throughout the body oftentimes do sorts of all these little tricks to increase surface area because they want to get more done. Like for instance, in your intestines, which are basically columnar cells, they look something like this. Normally, if you're absorbing nutrients, this is your working surface area here. Not a whole lot to work with there, right? You can get some nutrients in, but you got to do better than that, right? So what they do is instead of increasing volume, what they do is they manipulate that outer surface. And they kind of create these little ripples like this. So now all of a sudden, all of this is now working surface area. So you can absorb a lot more on all of these if you're basically manipulating your plasma membrane that way so that you can create more surface area. So that's basically what they're doing. Cells are doing that all the time. You see that all throughout biology is structures are designed to basically increase their surface area. So now when you take a look at also the studying of cells, we have to talk a little bit about, because we saw some images already, about the main tool for this, right? The microscope. So basically when you take a look at the microscope, we have 2 main types of microscopes that we look at. Is the light microscope, which is oftentimes the one we use in lab. And we also have the electron microscope. So what the light microscope basically does is it essentially will use these glass lenses, kind of like magnifying glasses in series, and then it'll use light to beam itself on an object to view images. And so you can view these different specimens. They can be alive in there. They can be moving around. But typically what you're doing is you're increasing resolution. Now, well, that's really what you're doing with microscopy. But the question is, what is resolution? Well, resolution basically is your ability to discern two points. So oftentimes, one of the things that I do in a class is I'll do something like this, and I'll pick on you in the back. I'll do something like this. I'll say, okay, can you see those as 2 separate points in the back? Yeah, so you don't need, that's pretty good resolution. Now what I do is I get them a little closer together. can you still see 2 points? It's getting a little trickier, right? Depending how far away, it may start to look more like a dash. Some of you guys in the middle, it's like, okay, yeah, I can still see 2 points, but they're kind of close together. Now, what if I do this? Can you see 2 points? Now starting to look more like a blob, right? So what I did is I found the limit of your resolution. So what magnification does, it basically makes this bigger so that you can differentiate between those 2 points. That's what makes an object clear to you, okay? And that's what resolution is. And so in microscopes, in light microscopes, what's called the resolving particle is a photon of light. And this will work as long as your sample is larger than your resolving particle. So for instance, here comes your photon. What's going to happen is the light's going to hit the sample and it's going to reflect off of the sample, and you're going to see it with the assistance of lenses. Okay, this is how I see you guys. Technically speaking, I'm not actually seeing you physically, mass-wise. What I'm seeing is a reflection of you, right? So that's how I see you. It's a reflection of the light bouncing off of you because you are bigger than a photon of light. This only works if your sample is bigger than a photon of light. And so what we end up getting is this image. Now, with electron microscopy, it's a little bit different, right? Because let me do it right here next to it. So the question is this one. So this is like light microscopy. In electron microscopy, use an electron as the resolving particle. Because here's the problem. What happens if your sample is smaller than a photon of light? Let's say now this is the photon. When the photon comes in, it's not going to be able to reflect off the sample because it's going to swallow the sample up. And so you're not going to be able to get any view, any sensation of this because it's just too tiny. So you've exceeded the resolution limit of light. You need something smaller as your resolving particle. Of course, there's nothing smaller than an electron, right? Because it is vanishingly small, very tiny. So that's basically what it looks like. So if you take a look at different types of resolution, so for instance, your naked eye, you can see down to about 100 micrometers or about 0.1 millimeters. That is, if you take a look at a metric ruler, you can see a centimeter, which is roughly about the width of my thumbnail. A millimeter is about 1/10 of that. It's like a little thin line. 1/10 of that is about what your naked eye can see. It's about the equivalent of a speck, right? Just like a little point, a little speck, like a grain of sand. Okay, good. Computers. We don't see it with our eyes. It's electron microscopy. It's not like light microscopy where we can see it with our eyes. We visualize electron microscopy with computers. So the computers sense the electrons and then they generate the image. So yeah, because we can't see the electrons. It would just be invisible to us because our eyes can't see it because it only goes down to 0.1 millimeters. Right. So light microscopy at max magnification can get you down to about 0.1 micrometers. So a lot better. That's pretty good. Electron microscopy, like that's at 100,000x, can get you down to about 0.01 micrometers. Right. So pretty tiny stuff. So you can see very small structures. So this is what light microscopy looks like. So here you can see your red blood cells. Pretty good. You can see it's a pretty good structure. Like in anatomy physiology, a lot of times we'll be working with light microscopy. Now, one of the things we see here is basically stains, right? A lot of times in microscopy, we use stains will help us to create contrast. So we can see the sample, right? So it's not normally purple, it's not normally pink, it's usually translucent. It's clear, but we put stains in it to dye the tissue so that we can see it. That's what we use those stains for, is to give us a little bit of contrast so we can see it. This is a transmission electron micrograph, so you can see that basically a transmission electron micrograph is a situation where you take electrons and you beam it through a sample. So it's like a cross section of a sample, and then you basically view it there. And then a scanning electron micrograph, this is a little bit different. So this is a situation where you have your sample and you take the electrons and you basically glance them off of that. So you see a three-dimensional topographical view. So the scannings are like really cool. It's like what it would look like if you were to miniaturize yourself and fly in there, like Fantastic Voyage style, which is a classic 60s sci-fi movie. For you who are into classic 60s sci-fi stuff, right? Looking at you, Doctor Who fans, right? What, all like two of you, if any. I like Doctor Who, but I like the more current ones, not so much the 60s ones, because it's just like, okay, that's just like a garbage can with like ears on it. It's like the newer ones are interesting, but right. So basically then, you use these lenses to magnify the image, and you get photons of light bouncing off of the image so that you can see it. And so the combination of the two go together. Your transmission electron microscope will make a two-dimensional image of that. It's very high magnification, but the unfortunate downside of electron microscopy is the sample is dead. So you can't get a live specimen. You can see living things under light microscopy. You can see them swimming around in there. It's really cool. You cannot with electron microscopy. It's all dead. So if you're looking for an active process, you're going to have to do something else. So you're not going to be able to do things like time lapse or stuff like that, unless you happen to get like a lot of different slices, like in succession, kind of like you're taking multiple pictures and you kind of sew them together into sort of like this sort of flip book cartoon sort of a style. That's the only way that you can actually get something like that. But that's tricky. That's very difficult to do. So this basically will be projected onto a computer screen, and then we typically will analyze the image. Scanning is your 3D image. And so this is basically bouncing off of the structure. And so again, no live specimens, but you can get really nice structural information. So when we saw those red blood cells, those are all dead. Those are all dead red blood cells. They're not living. Okay. Okay. Now let's take a look at the organization. So basically, when you take a look at all cells in life, you can organize them into two main categories. You're either going to be prokaryotic or you're going to be eukaryotic. Those are your two, right? And so every single cell on the face of the planet falls into these two buckets. Now, how do you tell one from the other? So the way that you tell a prokaryotic cell is that, number one, is it lacks a nucleus. It doesn't have a nucleus to put its DNA in. And so generally speaking, that's the kind of the big one. Generally speaking, we have two different groups of these. We have what's called bacteria, well, this is a slightly different version of taxonomy. So basically, you have what's called eubacteria and archaebacteria. We oftentimes will hear those as bacteria and archaea. That's basically a slight variation on how we do taxonomy, which is changing all the time. Eukaryotes, on the other hand, have this really nice big nucleus. So for instance, in animals, plants, fungi, protists, things like that. Now, when we take a look at common things, let's take a look at common features of these cell types, not just differences. The things that all cells have, regardless of whether you're prokaryote or eukaryote. Number one, everybody has to have a plasma membrane. Plasma membrane is what forms the boundary of the cell. It's what basically defines the outer structure of the cell. Typically, they're going to be made out of phospholipids. We just saw that in the last chapter, right? So our phospholipid bilayer. And the phospholipid bilayer tends to be selectively permeable. That means it gets to pick and choose who comes into the cell and who leaves the cell, okay? Kind of a very similar sort of a way that you're kind of selectively permeable about who comes into your house and who exits your house, okay? So similar kind of an idea. So everybody's got to have one of those. Everybody has to have cytoplasm, right? This is that kind of a fluid substance that's inside the cell with all those things. And so there's different types of things in there. If you're a eukaryote, you'll have a lot of membrane bound organelles. If you're a prokaryote, you will not. You'll have different types of non-membrane bound organelles. So everybody has to have a cytoplasm in there. And so those are some common things. So this is kind of like what an actual cell looks like. Here's what a eukaryotic cell looks like. So here you can see the nucleus, which is this guy right here, that's your nucleus. This little guy right here at aggregation is what's called your nucleolus. We'll talk more about that in a second. The chromatin here, these blue patches, is basically where the DNA is. That's what the DNA looks like. Just kind of like ravels of DNA that are just sort of strewn out amongst the nucleus, doing its thing, doing what the nucleus does. And then out here you can see the endoplasmic reticulum, this kind of like labyrinthine looking structure. And then it's bounded, the nucleus is, by the nuclear envelope, which is a membrane system that basically defines the inner portion of the membrane. Now, this is a really, really good image. And this is oftentimes an image that I would highly suggest and encourage students to get familiar with. Memorize it, tattoo it on your body somewhere. Because a lot of times in a chapter like this, a lot of times students will be like, okay, wait a minute, how do I study for all this? Because there's a lot of structures coming at us. There's a lot of organelles coming at us, right? How do I basically structure? Well, this right here is a really great way to index the knowledge that you have in this chapter. Because what we have here is like a visual table of contents of what pretty much the rest of the chapter is going to be about, which is going to be about the organelles, their location, and their function. And it's a pretty common set of questions in a chapter like this. To ask a question like, what is this? And what does it do? Right? So that's structure, function. Like for instance, if I point to a mitochondria, which is this guy right here. So I say, what is this? And what does it do? Right? That's a pretty common set. So I always encourage students to take images like this, which has huge chunks of the textbook on it, where it sort of organizes that knowledge, number one. And number two is it's got a lot of information on there, right? So all those little detailed pieces of information allows you to be able to learn these things, not by flashcards in isolation, but learn them in context as they actually live and breathe and work in the cell itself. Oftentimes, these structures are easily understood when you see them at work in their normal environment. It's kind of like if I wanted to understand all the positions on a football team, I could take each one of them and flashcard them out. Quarterback, basically, the captain of the offense, or running back does this. Or the difference, like one that always gets me, between a tailback and a halfback and a fullback, right? So all those different backs, right? So I could flashcard all those and see what they look like. And I could do that for every position on the team. And I would have an understanding of what they were in isolation, but I wouldn't really understand how they worked until I actually saw a game and I was able to actually dissect. It's like, oh, okay, so here's what the running back is doing. Here's what the quarterback is doing, because he's doing it in context. It's not just the quarterback doing stuff. The quarterback is interacting with the running back, interacting with the wide receivers, interacting with the tight end, right? Interacting with the offensive line. And the offensive line is a lot of fun, right? Because the center is doing one thing, the guards are doing a different thing, the tackles are doing something different. And so understanding what they're all doing in their context allows you to understand what the whole purpose and the function of them actually is. Once you get to that point in football starts to get a lot of fun, because then you can sort of dissect out the offenses and sort of figure out the kind of plays that a team is running. And then in off season, it gets really fun, because then you realize it's like, we don't just need like a tight end. We need like a blocking tight end, because we're kind of run heavy. So, we need a tight end that can block, that can help out with the blocking team, or like a running back. It's like, we don't just need somebody who can run the ball. We actually need somebody who can stay in the backfield and take on those defensive ends as they come in the backfield to do some blocking, so that the quarterback can have a little extra time to get it to the receivers, those sorts of things. So you kind of start looking at like, oh, these are the needs, and these are the kinds of plays that we want to run. So we need these sorts of players. That's kind of what we have here, right? So we basically have this sort of context-driven way of learning the cell. This is what I would really encourage you guys to do. Okay. We're going to take a look at about a little bit of the origin. We're going to start taking a look at structure, right? So the idea, and this is kind of like one of those things where we take a look at sort of events that we always ask this question, right? Because we see this massively complex eukaryotic cell with all these different organelles in it, all sorts of stuff going on here. And of course, the natural question is like, how did all this stuff come together, right? I mean, this is like a lot of moving parts. There's a lot of variables here. So how did this all come together? So that's an evolutionary question that oftentimes we ask. Now, remember, as is the case oftentimes with evolutionary studies, is typically the further back in time we go, the harder it is to know things for certain, because a lot of the direct evidence and the direct data that we would normally be using has long since vanished and been annihilated through different geological upheavals that the Earth has gone through. So it's kind of like doing forensics without any evidence, right? So it's tricky. So as a result, we tend to have a lot of different versions of the narrative. It's kind of modeling. So we kind of model different ways that things could have come together. There's probably about a half dozen versions of these, because in a respect, we're talking about events that happened some 3.5 billion years ago. And it's hard to know for certain what happened 3.5 billion years ago. So we have like a half dozen different narratives that explains it. It could be something completely different. So that's always a thing. So I always say, a lot of times students will be like, but there's like a lot of different ways that there is, because we don't really know for sure how it all came together, right? We're constantly finding stuff that's like, well, this suggests that maybe this happened. And so we're constantly sort of editing and revising this. But here's how it worked. Back in the day, when the Earth had no oxygen in its atmosphere, when the Archaeans ruled the world, these are the extremophiles, right? Those prokaryotic organisms that lived in very ugly regions like the thermal vents, right? Those volcanic thermal vents, very dangerous, very noxious places to live, right? Very acidic, oftentimes, very hot, right? Normal organisms like us would have no hope there whatsoever. And then from this, basically, we get the idea of endosymbiosis. So endo means inside, symbiosis means it's kind of like I scratch your back, you scratch mine kind of a relationship. So it's mutually beneficial. And so oftentimes what we suggest is that basically there's maybe a couple of ways that this could have come together. So one of those ways is that perhaps back in the day, originally we had a prokaryotic cell with its DNA just sort of dumped in there in the middle. And then somehow, and we don't know quite exactly how, this we started to form invaginations like of the plasma membrane that started creating these flaps of membranes that then sort of encircled the DNA. Right, so that's one particular version of this. That creates our nuclear envelope, that creates our endoplasmic reticulum. And then this cell then, one day, gobbled up an aerobic bacterium, something very similar to mitochondria. But what we did was we realized that, wait a minute, we're not just going to eat you, right? We're gobbling you up, but we're not just going to eat you because you're actually making energy that we can use. So I've noticed that if I just feed you some molecules like glucose, you'll produce energy that I can use. So you're actually more valuable to me alive than dead. So what I'll do is I'll do this. I'll basically give you safe harbor. I'll let you live inside me. You keep making ATP for me. And I'll feed you all the glucose you want. And I'll protect you. So it's a little, I scratch your back, you scratch mine, right? I'll give you safety and you make energy for me. So that's endosymbiosis. So the idea is that basically led to ultimately the mitochondria. So some of those then would basically become animal cells where we have our mitochondria. Now others basically had that same thing. You saw mitochondria, but then you engulfed another bacterium, a photosynthetic one in this case, that made photosynthesis. And now you realize it's like, wait a minute. So what you do is something different. So I don't have to just go out and find glucose to feed my mitochondria, my little pet mitochondria, right? Now all of a sudden, I've got this guy who's a photosynthetic bacterium who actually makes glucose for me. All I have to do is expose you to sunlight and you're making all the glucose I could ever want. I don't have to go out and hunt for it anymore. You're making it for me. And I can take that glucose and I can feed that glucose right directly to my mitochondria and I can make energy. So guess what? You're more valuable to me alive than you are dead. So I'll give you safe harbor. I'll protect you. I'll expose you to all the sunlight you want. Make all the glucose you could ever want. And then I'll feed it to my mitochondria. My mitochondria will make ATP for me. And I'm happy. I got everything I want. Those became plants. Now, there's versions of this, right? This is kind of more of a gradualist version. There's also versions where none of it happened like this, right? Where we basically had kind of an archaea, which is where your nucleus came from. And then the archaea was the one that did all the gobbling, right? So it's a completely different trajectory So there's a lot of different versions. I'd say there's probably about maybe a half dozen different versions of the story. The reason why there's so many is because we honestly don't know how it all came down. Like it could be any one of those half dozen ideas. It could be something completely different, something that we haven't even considered yet. So that's kind of the idea of how we think it all kind of came together. Okay, now let's go ahead and start taking a look at some structure. So that's basically how a lot of those organelles came together. Now let's take a look at our plasma membrane. Functions of the membrane. So when we take a look at our membrane, remember it's mostly phospholipid bilayer. We saw that in the last chapter, but in this case, it's got a bunch of proteins embedded in it, right? So we essentially have a lot of functionality in there because the proteins themselves have a lot of function. Now, normally, when you put phospholipids actually in water, they will naturally create this bilayer. It's spontaneous. Matter of fact, my general bio teacher did this once. She took a petri dish of water and she had some phospholipids. And what she did was she injected the phospholipids in the water. And you know what happened? Like that. We couldn't even time it. It formed like a little soup skin on the top. That soup skin was the phospholipid bilayer. And so this happens like immediately. Like if I were to put phospholipids into water, it would immediately start to form this bilayer. Why? Because remember, this phospholipid is a very disturbed molecule, right? It's part polar, part nonpolar, which means it hates itself. Like the polar part of it hates the nonpolar hates the polar. And they're not just like, self-hate. It's like they want to eliminate themselves, right? It's like, I want you to stop existing in the universe. But at the same time, I love myself. But then I want to kill myself. But then I love myself. And you can imagine if you're going back and forth, you're a very fractured psyche, right? So when you dump these guys into water, what happens is basically the polar head is happy. It's like, hey, I got my polar water friends here, right? So I got my hydrophilic friends here, so I'm happy. But those nonpolar tails are very unhappy because they're like, oh my gosh, it's like I'm just like surrounded in water stink, right? I mean, it's just touching, it's all over me. Imagine for a nonpolar tail, it would be like as if your legs were on fire. Would you be motivated to put the fire out if your legs were on fire? Like literally, if somebody put gasoline on your pants and lit your legs on fire, you'd be pretty motivated to get that to stop, right? You would probably do just about anything you had to in order to get it to stop. Even if I had like a tub full of urine, which normally you wouldn't get anywhere near, better known as a honey bucket, right? You would probably jump into the honey bucket, urine and poop and everything, to get that fire to stop. Now, does that mean you enjoyed getting in there? No. But my guess is once you start feeling the fire starting to burn your skin and starting to work its way through the layers of your skin, you'll do whatever it takes, even if it means getting a little dirty in the honey bucket, because that's the only source of water to put out the fire. Those nonpolar tails are feeling the same way in water. So as a result, they are willing to do anything, which basically means what happens is they will automatically form this bilayer, which is designed to eliminate water. So they'll point their phosphate heads toward the water, and their nonpolar tails will cover each other and protect each other from the water. But guess what? These ones on the side still feel the water, don't they? So what do? They cover themselves up. Ah, that's better. But now these guys feel the water, right? So they just cover themselves up. But now these guys feel the water, right? You get the idea? When does it stop? How can you create a bilayer that's not in contact with any water at all? Yeah, you just form a circle. There's your bilayer with your little nonpolar tails all pointed in the middle. Guess what you just made? A crude plasma membrane, a cell membrane. And guess what you had to pay to get it? Nothing. It happens spontaneously just because of the nature of the phospholipid. And then ultimately what you do is you can basically modify that by plugging in all these different proteins. So you can see all these different proteins. You can see your bilayer first of all. So here's your lipid bilayer with your phosphate heads up here and your nonpolar tails. Here, it's like the No Water Club. Now, in order to stabilize your membrane, you have cholesterol interjected into the membrane itself. That kind of stabilizes the membrane from wild swings of temperature, right? Because lipids melt when it gets hot and they harden when it gets cold. So to kind of help buffer against that wild swings of structure and temperature changes, cholesterol is stuck in there because cholesterol is not the same structure. It doesn't behave in temperature the way that these phospholipids do, so it doesn't melt. It doesn't set up like grease, basically. It just basically stabilizes everything. And then within that, you start to interject all these different proteins. So you can see a lot of different proteins here. For instance, you can see an integral protein. This guy right here is probably some sort of a channel. So you can see it's creating safe passage for some sort of a molecule to be able to pass through. You can also see this is an integrated protein of one sort, but you can see a peripheral protein, which is not actually inserted into the membrane. It's actually associated with the membrane. You can also see things up here. These are glycoproteins. Basically, you see a protein that has this carbohydrate branch on it. And you also see glycolipids. So here you can see a lipid structure with a carbohydrate bound to the lipid itself. So the glycolipids, here's another one right here, and the glycoproteins are important because these guys are associated with cell identity. This is how liver cells sort each other out, how one liver cell knows that it's another liver cell, because all of your cells have glycoproteins on them. It's like a barcode. It's like you scan the glycoprotein profile and you're like, hey, you've got the glycoprotein profile of a liver cell. I'm a liver cell. So why don't we join team and start creating liver tissue? Right? That's how they organize themselves. Also on the outside of the cell, you'll notice that you see what's called extracellular matrix. So this is basically all the secreted proteins and molecules and things like that that get secreted on the outside of the cell that the cell creates for itself. Okay. A lot going on in a cell. So that entire image is basically kind of a visual of what we now refer to as the fluid mosaic model. So basically what we have is a fluid membrane with a mosaic of proteins inserted into it, like rafts on an ocean. Imagine a raft just sort of freely moving laterally, just sort of drifting about in this ocean-like lipid membrane. Okay, that's the idea of the fluid mosaic model. So you'll have cholesterol to stabilize the membrane. From temperature effects. And you have glycoproteins and glycolipids, which basically are cell identity markers. These glycolipids also allows your cells to distinguish self versus non-self. And they can act as receptors. So this is basically the basis of your immunity. Because every single cell in your body has like a name tag on it, right? Like every single cell in my body has a mark tag in it. So that's how my immune system sort of scans my entire body, checks everything out. If it sees my name tag, you're good, you're legit, you belong here. But if I happen to get like a liver transplant and I've got Bob's liver, when my immune system scans that tissue, it doesn't see my name tag on it, it sees Bob's name tag on it. And then that's non-self, so that doesn't belong in my body, so it attacks. And that's the basis of tissue rejection. Okay, there's a fundamental breakdown of that self, non-self recognition. Okay, so some of the things that you have at the membrane, so you got different membrane proteins. Some of them act as channels. For instance, this is basically allows you to allow some things in, but not everything. So if you're small and hydrophobic, like oxygen or CO2, then you can pass freely through the phospholipid bilayer. Why? Because these are nonpolar. Oxygen is nonpolar. Carbon dioxide is nonpolar. And the membrane is also nonpolar. So when it sees that, oxygen has no problem getting through because it's also nonpolar. So the channels, however, basically will allow polar molecules through. So if this is a polar molecule, it has to go through a channel in order to get through, but you have to build that channel, right? It's just like you're not getting through the mountain unless we build Eisenhower Tunnel. So a channel is like Eisenhower Tunnel. It creates safe passage for us to get through this obstacle, which is like the big mountain. Okay, so water, for instance, needs a channel. Anything that has charge to it, like ions, needs a channel. And this will allow them to pass through. And channels typically are specific. So what if you want sodium ion to get through? Guess what? You have to build a sodium channel. If you want calcium ion to get through, you have to build a calcium channel. If you want water to get through, you have to build a water channel. The water channels are called aquaporins. Okay, so they actually have to go through their own channel to allow them safe passage. Yeah. Do I? Yeah, because if you don't have channels, then nothing's going to be able to get in and out of your cell. And part of the thing is most of your cytoplasm is water, which means you need all those solutes and you need water to be able to move back and forth through your membrane in order to keep your water balance. And so if you don't have those channels in there, you're not going to have that water balance, you're not going to have that solute balance, and you're not going to be functional. So you'll essentially die. The cell will die. Okay. So some of the things that are happening in terms of some of the functions that we see at the membrane, some of the movements. So this is kind of like a table of contents. We're going to talk about each one of these individually. So we have diffusion, osmosis, we have what's called facilitated diffusion, active transport, and then endocytosis, exocytosis. So these are basically transports, different types of transport. This is a transport function. Of membrane proteins. So this is kind of what it looks like. So here you can see, here's a channel for water, the aquaporin. So you can see the water can move back and forth through the aquaporin. Ions cannot get through that membrane unless they have a channel for it. Small, non-charged, non-polar molecules are free to pass through, like oxygen and CO2. Large macromolecules cannot get through. So ions cannot get through. So you can see that basically you have selection. You can pick and choose as a cell what you're going to allow past your membrane and what you're not going to allow past your membrane. That much you do have control over, right? But if you don't allow passage, it's not going to pass. That makes sense? So basically, we start the entire conversation off with diffusion. So this is kind of like a little bit of a table of contents for this diffusion transport section. So we'll kind of go through all of these. We'll begin with diffusion, but the transport concept is a good topic. It's going to take a little while to unpack, and we want to make sure we kind of do it all as a block. So this is actually a good place to start since we're already at our witching hour.